Cell Signals and Communication by Abhishek, Om, and Keshav. We're going to start off by explaining the G-protein receptor system. This system starts when the ligand, also known as a signaling protein, binds with the G-protein receptor, which is embedded in the cell membrane. This results in the inactive protein with GDP attached to it to move and make contact with the G-protein receptor. After this, a phosphate in the cytoplasm binds with the GDP to turn it into GTP. As you can see here, the phosphate is moving up to go bind with the GDP. Once the GDP is turned into GTP, the protein becomes activated. This active protein goes to move and fit the inactive enzyme. Once the active protein binds with the enzyme becomes active and changes shape, which is transduction, and releases a cellular response signal, such as the release of calcium ions from the ER. Protein leaves the enzyme, rendering the enzyme and protein both inactive. This is the tyrosine kinase receptor system. This system starts with the two ligands going and binding with the two tyrosine kinase receptors, which are embedded in the cell membrane. Once these ligands bind, the two tyrosine kinase receptors start moving towards each other. When they get together, they form a dimer. 6 ATP molecules start moving to meet the 6 tyrosine receptors. When they attach, they each deposit one phosphate, and the remaining ADP leaves to go be used elsewhere in the cell. After the ADPs leave, two inactive relay proteins start moving towards the phosphates. The relay proteins become active when they bind to this phosphate. They then change shape and send two signal responses. They then detach from the phosphates and go elsewhere in the cell. After this, the ligands then leave their respective tyrosine kinase receptors. Now we're going to do heroin and its effects on cell communication. Heroin affects the way brain cells function through a person's feelings. Now we're going to show you a person's brain cell under normal conditions, which is feeling no emotion. The neuron that contains the inhibitory neurotransmitters are released and bind with the receptors that block the release of dopamine neurotransmitters, which make you feel happy. And a person's brain cell, when they're happy, does the exact opposite of this. A native opiate binds with the opiate receptor, which blocks the release of inhibitory neurotransmitters, and due to this, these inhibitory neurotransmitters cannot block the release of dopamine neurotransmitters. These dopamine neurotransmitters are then released, and they bind with the dopamine receptors on the neurons. And every time a dopamine molecule binds with its receptor, it creates a stimulated reaction in the neuron. And this is a person's cell under the influence of heroin. It simulates the feeling of your happiness. Rather than an opiate binding with the opiate receptor, a heroin molecule binds with this opiate receptor. So this then blocks the inhibitory neurotransmitters from being released and releases the dopamine neurotransmitters instead. Heroin is used to create a fake feeling of happiness. And again, as when you're feeling happy, when these molecules bind with the dopamine receptors, they create a stimulated reaction of happiness. Now we're going to analyze where the breakdown in cell communication occurs due to the use of heroin. As you can see, the pivotal moment is when heroin binds with the opiate receptor. This blocks the release of inhibitory neurotransmitters and lets dopamine be released, and this in turn makes you fake the sensation of happiness.
Some symptoms of heroin include dry mouth, constriction of pupils, false sense of happiness, itching, nausea, and skin infections. Treatments of heroin usage can be medications such as methadone and naltrexone, which can help with the physical aspect, while doctoral counseling can help with the psychological aspect of getting over heroin.